Welcome to the Protocol Particulars series. This is a video interview series where we're connecting with researchers that are part of the ASAP community in order to learn more about protocols that they're depositing in protocols.io and making available to the rest of the scientific community. I'm Devin Snyder, and I am the host for today's episode. But with me, I also have Francesca Tonelli. Francesca is a senior scientist. She's part of the Dario Alessi lab at the University of Dundee, and she's part of the Alessi team for ASAP. And on the side is also the project manager for Team Alessi. So she has a lot on her plate. Hi, Francesca.
Hi, everyone.
For today's episode, we are going to talk about your protocol, the quantitative immunoblotting analysis of LRRK2 signaling pathway. I'm pretty excited about that. So for my first question, I actually just want to give the users some context about ASAP and what Team Alessi is doing. So I'm wondering if you could just tell us a little bit more about your ASAP project.
Yeah, sure, Devin. So our research focuses on the leucine-rich repeat kinase 2 or LRRK2 signaling pathway, as mutations that increase the kinase activity of LRRK2 represent one of the most common familial causes of Parkinson's disease. And the goal of our project is to understand how the LRRK2 signaling pathway is regulated and how a dysregulation of this pathway might result in PD initiation and progression. So ultimately, this may lead to the development of novel approaches to better diagnose and treat PD. So our project relies on the ability to robustly and reliably monitor LRRK2 activity in cells and tissues in response to specific stimuli or genetic mutations. And this can be achieved using various techniques, and immunoblotting is one of these. In particular, we monitor the phosphorylation levels of LRRK2 substrates like RAP10 and RAP12 by immunoblotting as a readout of LRRK2 activity in cells and tissues.
Thanks, Francesca. So it sounds like there are a few different protocols that can be used for this, but I'm wondering if this protocol was originally created by your team or if there had been other protocols that were previously established for this immunoblotting, and if there had been previously existing protocols, if those just didn't work for your team for some reason, and why you maybe needed to modify those if they had existed.
Right, so Western blotting or immunoblotting is a well-known method which dates all the way back to 1979. And with this method, specific target proteins are detected within a complex mixture of proteins extracted from cells or tissue samples through their binding to specific antibodies. Now, with time, we've come to realize that while our research group considered this technique as a routine technique, this was not necessarily the case for other researchers that are working in the field. So many research groups have contacted us over the years for troubleshooting, asking how our lab would consistently produce beautiful, clear-cut immunoblotting data. Hence, the motivation to publish a detailed step-by-step protocol that would enable others to reliably measure the expression and modifications, such as phosphorylation, of the LRRK2 signaling pathway components. And we strongly believe that there can't be open science without open methods, and hence the choice of protocols.io.
So you mentioned that groups have been troubleshooting in the past. Was there anything that your group had tried that didn't work, that didn't make it to the final publication, or did you guys have to troubleshoot it all while you were creating this protocol?
I was fortunate enough to inherit a very good protocol from previous members of the lab, but there are some critical steps that, based on the troubleshooting requests that we received over the years, I like to highlight. So the first critical step is the cell lysis or tissue lysis. So first, it's absolutely critical to add protease and phosphatase inhibitors to the lysis buffer and to keep the lysates on ice at all the time to protect proteins from degradation and preserve their modifications, such as phosphorylation. with regard to which detergent to use in the lysis buffer, we find that 1% Triton X100 is the optimal detergent and concentration. However, the lysis conditions are slightly different for some applications, for example, in co-immunoprecipitation experiments. And for that, we have written separate protocols. Finally, the volume of the lysis buffer is important. If you use too little, proteins won't be efficiently extracted from cells or tissue, and this will impact the readout. Now, another critical step regards the method of protein transfer or blotting from a gel to a membrane for subsequent antibody probing. So we find that wet transfer is the most efficient transfer method to detect the LRRK2 pathway components specifically. Something else we noticed is important is the pH of the wash buffer. So I will make a note just to make sure you double check the pH of the buffer. And antibodies. So the quality of the antibodies used to detect the proteins of interest plays a significant role in the success of this technique. And I would say, do not assume that all antibodies that you find out there are good, specific antibodies, which brings me to my next point. Always use positive controls. I cannot emphasize enough the importance of including positive controls in your experiments. This will enable you to assess, for example, whether the antibodies you're using are actually specific for the protein of interest, but also whether your immunoblotting technique is as good as it should be. One last thing I would add is that it is okay to make changes to a basic protocol like ours. Obviously, there will be variation to this method that we haven't tested, but may work as well. Once again, remember to include positive controls, as this will enable you to assess whether the changes that you're making to the protocol are somehow negatively impacting the readout.
Thanks, Francesca. That is a really good list of all the critical steps that are part of this protocol. So you listed out all these critical steps. Of those steps, are there any that are particularly tricky that other people might make a mistake on?
I would say it's important to take the time to measure the protein concentration of the lysates and also take the time to load the samples onto the gel. Do not rush that step. And finally, take the time to set up the transfer properly to avoid those dreaded bubbles. Anyone who's been doing Western blotting will know what I mean.
Yeah, I know what you mean. Those are the worst if you have a really great blot and a bubble shows up, and it's always right where your antibody is going to be binding. So those were all the questions that I had prepared for you. I'm just wondering if there was anything else that you would like to share that I didn't ask about.
Yes, just one last thing. I would recommend reading and using our protocol just as a good general immunoblotting method and not just necessarily specific to the LRRK2 signaling pathway.
All right. Thanks, Francesca. This was really helpful, and I'm sure the rest of the community will really enjoy watching this.
Perfect. I'm glad.